exactly. I think Manoj has also joined in. I cannot really.
Hi, nice to
Yeah,
meet you. Thank you for all the
yeah,
emails, and I'm
yeah.
sorry for the confusion. I put you in my calendar at 10 to make sure that I didn't get overbooked and wasn't late. And then I was like, oh, panicking because we have an exam today. It's all
No,
good.
no
So
problem.
thank you for the
It's
understanding.
really a big pleasure to that you are here. And Manoj probably has already told you what we do. I
A little
think,
bit, yeah.
yeah. I mean, I think also I feel a bit obligated to sort of say something from my side as well.
Sure.
Um, so I am very generously being, let's say, given the job by Manoj to be the chairperson of our organization.
No. Actually, only because of this person, I'm facing you. Only because of this person.
So, so that's why we work together since a long time, and what we do is that we have this platform which is associated with NGO, a nonprofit which Manoj actually runs.
Mhm.
And we are trying to reach students and professionals in science, especially in biology, and making them aware of research globally. And because we believe that research is global, not national, so we should collaborate globally and
Mhm.
work globally, take experiences from in a global manner. So I think that's what we believe in. And this is an effort in sort of starting up this organization. We have now talked to quite a lot of scientists. I think this is sort of our first phase where we are making people aware of who we are.
Mhm.
And this by basically talking to people. We do have plans to organize courses, seminars, in the future, maybe conferences if everything goes well.
Yeah, we are also
Um,
planning a conference in India.
yeah,
Oh,
yeah,
great.
yeah.
Yeah.
So this is sort of what we do. We we also want to reach out to people in basically all corners of the country in India, so that people know what the global scenario is. And I think this is really, a really an big effort from Manoj, who has started
Um,
this idea, and he's reaching out to everyone, which really gives me
no,
gives me pleasure to see that, because I mentor him in what he does.
no,
So
actually, let me clear one thing, ma'am. Actually, this person means Sudip means he told me how to approach, means what is the approach in science and how to communicate. Uh, my English is not too much good or not too much bad. I'm middle, below average in the English speaking. But this person teach me how to communicate. My communication is too much good. I'll communicate with any other person, and I'll approach. My approach is going a good way, and he or she might be say yes sometimes. Means from a small town village to India top place like in IIT, only because of this man. And right now, I also got an opportunity from Europe.
yeah,
In,
okay, yeah. So that's basically who we are. I think there's a big team behind us as well,
Mhm.
and they all help us in different aspects, starting from social media to PR campaigns to reaching out to people, setting up all these meetings and stuff. So a little bit about me. I am Sudip. I am right now a staff scientist at the Technical University of Munich in Germany. I'm
Okay.
actually sitting in one of our buildings with an open dome,
Yeah, it
and
looks lovely and bright there.
yeah, exactly. I am so my training is an infection biologist,
Okay.
and I have worked in did my PhD in Germany as well. Worked in different places. Also did my postdoc in Switzerland,
Mhm.
working in microbiome science, and now here I am basically responsible for all the projects in the wet lab aspect of our chair for microbiome science, human microbiome. So this is who I am, and you know now about BioFolk. But I think let's get started to a little bit more
Okay.
about you. I think
uh,
the best premise would be you tell a little bit about you, who you are, and your journey has been.
Yeah, sure. Well, first of all, thank you very much for the invitation to join you today for this sort of chat, I guess. So I'm Cleo. I'm a professor of senescence at the Blizzard Institute, which you can see behind me here. And we're based in East London, in the UK at Queen Mary University of London. I've been at Queen Mary's for a long time now, maybe longer than some of the people watching this might have been alive. And I've been a biologist, I think, since a very early age. I was fascinated with biology at high school. And so I feel very lucky that this is my day-to-day, my job, as it were, vocation really. And my lab at the Blizzard is interested in the process of cellular senescence and trying to understand it in the context of aging and cancer.
Okay, nice. I think maybe Manoj, I start with a couple of questions. I think students will be really happy to know, from an expert like you, what it is, what are the concepts. So I'm going to probably ask you a bit about basic concepts. I think that's what we should start with. And I think we've all come across senescence in cell biology, whoever has learned cell biology. I mean, I remember that I did. But could you then help us understand what exactly is cellular senescence and why should we care about it?
Well, I guess what is cellular senescence? I think we're still trying to understand that as a biological concept, as a mechanism and as a cellular consequence. But I guess put simply, it was originally discovered as a way in which our cells potentially age. So senescence is a Latin word which essentially means to grow old, and I think we've adopted that to describe this cellular phenomenon. But actually now, through the work of many groups, we understand that senescence is a fundamental cellular program where cells can respond to a range of different pathological, physiological, extrinsic, intrinsic signals. And they make a choice to enter this stable, durable cell cycle arrest where they remain metabolically active. Their metabolism can change, but they essentially are still functioning in a certain way. But they undergo a number of really important changes, including the molecules that they secrete into the local microenvironment, which can have beneficial or detrimental consequences depending on a range of different factors. So it's a fascinating, enigmatic, complicated cellular process that we're still really trying to understand. And why we should care about it, I guess, there's probably two key reasons why I think we should care about senescence. One is that it's a really important, well-established tumor suppressor mechanism. So it's a very important way in which our cells can defend against cancer evolution. And understanding how they do that, why they do that, and what that means in the context of cancer evolution, I think is really important. The other reason why I think we should really care about senescence is some really landmark research from a number of different groups, really spurred from an original observation from Darren Baker, over 10 years ago now, where he found that if you genetically cleared P16 positive cells, so P16 is one of the effector molecules that can engage the senescence phenotype, if you remove those cells from a mouse, either short-term or over its lifespan, you could fundamentally change the aging trajectory and a number of physiological features of the mice. And so this work really positions senescence as a causal, as having a causal role in aging. And you don't need to, pick up many newspapers or read many journals these days to know how important aging is and how much of an important factor our aging population is for countries across the world. And so if we can understand what senescence does in an aging context and try and influence it to help us live healthier for longer, I think that could be a really neat thing to be able to bring to communities and societies around the world. So I guess those are the two main reasons why I think everyone should care about senescence.
I think, it's actually, I'm not at all a person who understands cell biology that well. I guess I understand apoptosis, necrosis, cell cycle, but senescence has always been a bit of a let's say a tricky area to understand. I'm sure that you also agree that it's not that easy to study.
It's really not. And actually,
yeah.
the more we understand about it, the more we realize we don't understand it.
Yes, I think then let's talk about a bit about your work. I think what I've understood is that I think I just briefly had time to see that you had a study in 2024 where we were talking about senescence markers, I guess. That was and could you then shed a bit of light on what your lab does in this?
Yeah, sure. So my lab is obviously interested in senescence.
Yeah.
And we look at it in both the context of cancer and also in the context of aging. So maybe if I briefly summarize the cancer work. So we're very interested in trying to see if we can harness senescence as a novel anti-cancer treatment. So our kind of hypothesis is pretty simple. If we can take a cancer cell and we can drive it into senescence, into this stable, durable cell cycle arrest, maybe that can do two things for the tumor. One, it can stop it growing, which is hopefully a good thing. And two, we're taking it into a fundamentally different cellular state, which means that it might be susceptible to a whole new class of cancer drugs or drugs that could influence the tumor trajectory. And so we do a lot of work trying to understand how we could do this in a cancer setting. So first, driving the tumor into a senescent state, which is kind of referred to as the first punch of a 1-2 punch strategy. And then we try and find a second class of drugs in which we can kill the senescent cells, so activate that process of apoptosis that you referred to a moment ago, Sudip. So we have an active drug discovery program, I suppose, in that area. And at the moment, we have a molecule which is currently under evaluation with a pharmaceutical company who is interested in cancers that women have, and particularly in this context, we're looking at a type of breast cancer. So that's one half of the lab's research. And within that, we try and understand not only the drugs that we could kind of discover, but also the cellular and molecular mechanisms which might drive this process. So what is it that we're doing to change the cell to make a cancer cell senescent? And what are we doing to it to try and kill it in a new route?
Okay.
And then the other half of the lab is interested in the role of senescence in aging. And really for that, we're trying to understand what are the kind of physiologically relevant triggers of senescence that occur during aging. And for that, we look at the interplay between aging hallmarks. So I'm not sure if you're familiar, a bit like the cancer hallmarks, there are a number of different aging hallmarks, one of which is cellular senescence. And we're very interested in asking how different aging hallmarks interact with each other. So we have programs looking at the interplay between senescence and epigenetics, senescence and altered secretome, senescence and proteostasis, and so on. And we do our work in that context using a range of different approaches. So quite often we use unbiased phenotypic screening as a means of trying to understand these processes, to see what mechanisms we can uncover. We have drug discovery programs in these areas as well. And we do a lot of work with high content image analysis. So really trying to understand what's happening at single cell level using either microscopy, but in the high content sense, but also techniques like single cell RNA-seq, where we can really look at signatures at an individual cellular level.
Yeah, that's actually cool, because I saw that you really integrate sort of the quantitative biology aspect of it, and also RNA-seq, single cell RNA-seq, omics data. So was that always a choice, or was something of a necessity for you to use those techniques, and you have quite well integrated these two, I guess?
I think it's probably both a choice and a necessity, because the more we understand about senescence, the more we appreciate that it is a cell type and context dependent phenotype. And looking at stuff at the bulk level can be very helpful. A lot of people have done really great studies looking at bulk RNA-seq, for example, to see what how the transcriptome is changing when you apply a particular trigger. But actually, when you start to drill down as to what is happening at the single cell level, you realize it's really complicated. And so being able to use microscopy and single cell approaches really helps us to, I guess, get a new perspective on those changes, and it can lead to some really interesting observations around subpopulations and heterogeneity, which I think is really interesting.
I've also worked a bit on the single cell aspect from the immunology side. And I think it's really, we were trying to detect surface antigens in a single cell level, and so that was really, I think it's really gives you a whole different fresh perspective on the heterogeneity of cells. You would expect a bunch of cells in a cell culture sitting there must have the same thing, but it is not true. And it's very fascinating. And I actually really like those kind of data, I think it's really nice that you've taken this very classical cell biology, which people studied for decades, and fused it with something at a systems level. I think this is really, really cool. Before I think Manoj next to take over for a few questions, but I would like to ask one more question before that. Tell me a bit about the institute itself, where you are, about the science, about the students, about how people get an opportunity to work there.
Yeah, sure. So I don't know how much you can see behind me. So this is part of our institute. It's architecturally designed research institute. So it is. It's quite unlike anywhere I've worked before. And if you have a chance to just have a quick look on our YouTube channel, we have a great drone video of it kind of driving around the building. Yeah. It's all open plan. It's really modern. I don't know if you can see just behind me in the top corner here. We have a number of suspended meeting rooms, which were kind of in sort of a fusion of art and science and engineering. So that is the mitochondria behind me.
Oh, nice.
We have a really fantastic award-winning public engagement pod just to my left down there. And so what that enables us to do is to have local school kids come into the institute. And so we're one of the few institutes that has something like this established. And inside the pod, there are a range of video games and interactive sessions that the kids can do to find out about the research that we do within the institute. And so it's really nice when you have these kids walking over the bridge and they get to see down into the lab, see us all in our white coats. And so we have that as something where we have regular visits from local school kids. And it's a really nice way of engaging with our local community. And also a great opportunity for our PhD students, our postdocs, to practice their public engagement skills, because it's one thing chatting like this about cell biology, but if you're trying to explain your research to someone who's 7, you need a different way of communicating and explaining and engaging with different groups. The institute itself is very I guess diverse in what we do, the kind of areas that we research in. So we have 5 different centers. So a cell biology center, which has an interest in skin biology. We have an immunobiology department, a neuroscience department with trauma as part of its interests, and a bone and joint department. And actually just across the road from me is one of Europe's largest hospitals, the Royal London. And so we have really great links between the researchers here and the clinicians in the hospital. It's a short walk for us to pop and get clinical samples. So actually we do a lot of different types of clinical research within the building, a whole range of different areas. And I forgot one department, I knew I did, which is our genomics and child health department. I was thinking there's one more. Sorry,
I mean,
ECH.
with the sequencing aspect, kind of,
Yeah.
sure.
Yeah.
Yeah. No, great. That's very good to know, and I think a lot of people watching this would be very interested in. I'm sure they were very interested in working there as well. Maybe
Yeah, it's a
Manoj
great
takes over.
place, and actually the open planness
Yeah.
really brings a different. We were talking right at the beginning how collaboration and interaction is really important. I think the layout of the lab really does foster that kind of cross-pollination of ideas, because you're not in little rooms, you're in this kind of, this huge open plan lab with 400 other researchers working on all different areas of biology. So yeah, it's a really great place to work.
Yeah, great. All right, then Manoj, let's go for your questions.
Thank you. I would like to ask you, you are transitioning your background in cancer biology to cellular senescence, and what is the spark shift your research direction?
I'm sorry, the line is not great. Could you repeat the question?
Yeah. Means you are transition your background in cancer biology to focus on cellular senescence. What is the spark? What is the spark that shift your research direction?
Got you. I guess it was one of these kind of random interactions. So I went to a seminar, and so I would super encourage everybody to continue to go to seminars. I know it's hard to find the time when you're kind of busy in the lab, but I went to a seminar that was given at my institute when I was doing my fellowship at Imperial, which is sort of on the other side of London. And the seminar was by a man called David Beach, who's a fellow of the Royal Society, which is a very kind of high academic accolade in the UK. And he was one of the people who was instrumental in discovering P16, that key effector that I mentioned a little while ago. And he came to tell us about his research, and he just gave the most fascinating talk. It really piqued my interest. But at the time I was, a young postdoc. He was this senior eminent professor. But at the end of the seminar, the host said, we have one spare slot, if anyone would like to meet with David. And so I guess I was on this kind of wave of inspiration. And so I took the slot. I sort of said, could I have the slot? And so I met with David, and it turned out I was coming towards the end of my fellowship, and he was recruiting for a postdoc, for which I interviewed for and was fortunate enough to get the position. But it was really because of that seminar kind of shifted the direction that my research took, and actually, the area that my lab now works on was, I think, because of that moment. That spark, as you say.
That's the spark. Yeah, it should be like that. Yeah.
Then, what is the scientist you are admiring? Hello?
Sorry, could you say again?
Who's the scientist you are admiring most?
Which scientist do you admire the most?
Thank you. I think it's I don't know if it's just your line. You're very kind of fuzzy. Which scientist do I admire the most? I don't know, actually.
Would it be past or present, Manoj?
It's a tricky one, actually. The one that does come to mind is somebody who we lost in the field reasonably recently. So she happens to be a woman, which for me is, when I was doing my PhD, there was one female professor in my department. So you look up to these role models in a slightly different way. Her name is Judy Campisi, or Judith. Judy, we call her Judy. She was just I don't know quite how to describe her. She's a kind of very petite lady. She was a dynamo, but she was kind and gentle, inquisitive and collegiate and humble, and she loved to dance. She was one of the first, go to conferences and she would always be on the dance floor. So I think she just embodied a lot of really positive characteristics. as a human being, but also as a scientist. So yeah, we miss her. And yeah, I think the other thing that was really great about, I mean, there's so many good things about Judy, but one of the things that was great about her was that she could talk to anybody about anything, which I think was, really, really great. And she took a lot of interest in the young scientists in our field, which I think really is inspirational. That's really helpful. It's the young scientists that are going to come on to be the next leaders, and fostering our younger scientists is really important. So yeah, I have a deep admiration for Judy.
Yeah, I think that's a great answer. I think because oftentimes when we ask such questions, I think your answer has been the most unique until now. Because and thanks for sharing a personal story, because we always people would answer greats in the field, Albert Einstein, or I mean, of course, they are valid answers, but I think your answer was really
it's a
unique.
kind of emotional also.
Yeah, that's really nice. Yeah.
Then my next question is, what inspired you to start the phenotype senescence facility earlier on, and how has leading it start your aging cell research?
I'm sorry, I can't hear.
Actually,
No problem. I think Manoj meant is that what inspired you to start the phenotyping facility, the senescence phenotyping facility, and how is it until now leading that?
Helping your research.
Yeah.
Okay, all right. So the phenotypic screening facility, this was a little bit of a surprise to me. So I mentioned David Beach a few moments ago. And so I joined his lab as a postdoc, and he was setting up, this was a new institute when I moved here, only opened in 2005. And so he was creating a new group in the building. And one of the things that he tasked us with was establishing this technology. And so this was at a time when sRNA biology was really just coming into sort of mainstream. We were just being able to do genome-wide genetic screens. The libraries weren't even available when I started. And so David had this ambition, this aspiration to set up this technology in the institute, because he could see the power of being able to knock down every gene within the genome one by one and ask what happens in an unbiased fashion. So I guess it was his inspiration that had us set this up, and it was really fascinating, actually, because it was a great opportunity to learn about a whole new technology that I knew nothing about. We incorporated robotics, automated microscopy, and obviously these kind of cutting edge, brand new to the market, genome-wide libraries, sRNA as they were at the time. CRISPR wasn't a thing. So I think it was him, but it's been a real gift, actually, to have been involved in the establishment of the facility, and it's a continual pleasure to be still involved with the facility. So I remain the academic lead for the facility. I have a fabulous team who are the real experts now, who run everything in the facility. But what it means is I get to kind of peek in on projects that aren't to do with senescence, but using this technology to explore different areas of biology and medical science. So yeah, very fortunate to still be involved and still trying to develop and advance the technologies that we have within the facility.
Okay, really good. I mean, I think these kind of platforms are super important. I think it helps a lot. It takes a lot of time to establish, but I think it then solves decades-long problems after that. I think this is really an important facility.
My last question is that if you are not scientist, what would you have been?
Oh, that's a great question. So I think I would have I mean, I don't know if I would have been any good at it, to be honest. But when I was doing so in the UK, we have something called A levels, which is the last 2 years of high school. So 17, 18 age. And at that point, you have to really kind of decide on what subjects you want to study. You get to study 3 subjects, and that's kind of it. And so I was very torn at that point, that decision point, as to whether I would do biology type things, biology, chemistry, maths, or whether I would go down the art route, because I really loved art and drawing and anything kind of that involves any kind of painting. And so it was a tough choice, but I kind of did end up choosing biology, obviously. But yeah, I think that would have been the alternative strategy. And I still have this kind of backup plan in my mind that if, you know, it all goes to pot and I end up with no job, that I have a business idea which is called Cleo's Cards. And I will kind of make cards for people. I still do that now. I still make my mom birthday cards and all that kind of thing. And more recently, I've probably in the last kind of maybe 10 years, something like that, I've really got into doing pottery. So working with clay and building things. So I think that would have been the alternative. And I feel lucky that I still get to do that in my spare time. So I guess I've got the best of both worlds.
Really great. Yeah. I mean, I think, I'm also drawn to art. I was fairly good at it. I don't know how well I would be now, but I remember my sister used to be better at it. But I think it's really nice. I think the card idea is still, I think, a valid and a very viable business idea.
It's there as a backup. I've got to, you know, I have tried to do the maths as to how many cards I might need to sell to be able to make a living, which is a lot. So yeah, and I genuinely don't know if I'm any good at art, so we'll see.
Good. Maybe I take over a bit. I wanted to ask you about your advice for young scientists, and specifically students at master's or bachelor level who want to, let's say, pursue field of senescence or cancer biology. I think right now it's very difficult to draw a line between any of these, but in general, what is your advice to them? How should they proceed? What are your thoughts?
Well, my top tips.
Yes.
Well, I think my first top tip is and I know it's difficult, is to try and get some experience in the lab. I was very fortunate that I was able to spend my summers at university volunteering in a lab. I would work at the weekends and evenings to make rent, as it were. But during the week and during the day, I was lucky that people took me on as sort of a summer intern. And that did a number of different things for me. One, it helped me kind of work out whether research was a good, whether I enjoyed it or not. It's not for everybody, that's okay. And I found that I really, really liked it. I really loved it. And so I think that helped motivate me through my studies. But it also helped kind of guide me as to the research areas and also the techniques that I thought were interesting and I wanted to try and incorporate into a potential, research career. It also kind of started me thinking on how to design a good experiment and also how to critically appraise the literature. And both of those are kind of the pillars of being able to do good science, aren't they? So I guess getting involved in that as early as possible really paid dividends, I think, for me. It's hard and it's competitive, I know, to get these internships. And I get approached by students and I can only really take one at most over the summer just to support them properly. I have a student with me this summer, Joel, who is, been doing a great job. I think that would be my key piece of practical advice. The other thing I would say is to be curious and follow what interests you, because you both know science is it's not easy, even at the best of times. You know, even when everything is working, it's still hard work. It's still challenging. And if you're not curious and interested and fascinated by what you're researching, that hard work becomes really, really hard work.
Yes.
Even when things are going well. And when things aren't going well, and we all know what we mean by that, you know, you're doing an experiment and it's just not working, or something goes wrong. If you don't have that curiosity, there are those days when things really work and you have a eureka moment, or you have a result where you're really excited. They are not often enough that if you're not interested, I think curiosity is super important. And then it helps you think about your research as well. If you're curious about it, you're really pushing, you know, intellectually around the topic and your experiment and your ideas, and you know what questions you want to ask.
I think those are obviously like the pillars of it. I think a lot of people do know it, but somehow I feel that the students want something extra out of it. So I always tell them to upskill themselves, add more skills, I think, in the field of science. Or it could also be public outreach. It could also be anything, because science, we do science and we have to communicate to the society. So anything in the intersection of society and science could also be one of those things where they could gain skills. And I think, what do you think? So upskilling versus, which is more important? Upskilling is important, or getting more experience, which I don't necessarily think is the same thing, because we
they're
can
not
upskill.
The same thing. You're totally right. I totally
So
agree.
what are your thoughts on that?
I guess it depends what the purpose is. So if you're, I think let's take your scenario of somebody who's in a bachelor's degree in their first or second year of a 3-year degree. At that point in their career, they have, you know, 100 options open to them at the end of their career. Thinking about an academic research career is just one of the many, many, many things that they could potentially do. And so I guess when I was talking about getting research experience, it was really to try and use that as a vehicle to work out whether that is one of the avenues that is of interest. Because you have all of these things that you could do, not just one thing. And so you kind of have to play this delicate balance between working out, you know, because you've got to narrow it down somehow, haven't you, as to which path you want to take after the degree. And so I guess doing a combination of the two that we're talking about, kind of getting experience, but also getting some skills that will help bolster your CV when you're thinking about applying for the job, you know, whatever it is after you finish. Both of them are kind of equally important and equally valuable. And upskilling can also do the same thing for you. So you do some public engagement, for example, to take your scenario, and you find, actually, I love talking to 6- and 7-year-olds. It's brilliant. You know, this is the best thing. Or you're like, this is just not for me. So you do it and you can develop those skills, but you can also simultaneously decide, well, actually, of the options, I'm going to rule this out because this is, you know, this doesn't excite me. This doesn't bring me joy. This doesn't make me feel fulfilled. So I guess there's both. And actually, when you speak of upskilling, I think perhaps something I didn't mention in the context of kind of getting research experience, I think is very valuable. There's a lot of research now, and I'm sure you both find this, is a combination of wet lab, but also dry lab skills. And so getting dry lab skills is something you can do for yourself independently. You can upskill yourself in that area. And actually, I think it's something where having some form of bioinformatic type expertise is almost, particularly at the end of, say, a master's, we're almost expecting to see that. And certainly at the end of a PhD, if you come out of a PhD without some level of dry skills, you know, so that's something you can do for yourself. And there are there's a lot of very very good free tutorials, data sets that you can download and, take through the process for yourself. And that can kind of fulfill two things, can't it? It can give you a bit of research experience, but it can also give you those upskilling points, as it were, that are very helpful. And it shows initiative, which I also think is always good.
Exactly. Yeah. I think this is really important. I also say that, I mean, it can be as simple as, for example, I now really feel how useful it is when I started doing my PhD. One day I thought, hey, I would not use Excel anymore. I would use R.
Controversial.
Yeah, I would use R. I would not use Excel anymore. And people were like, really? And I really made sure that until I wrote my thesis, every single plot in there was made in R. And I really did that. And this was something which I did by my own. And now I feel that it helps me do right from making a plot to RNA-seq data analysis. So this is sort of what I also tell people, that you have these skills which you can pitch in front of someone.
It's
really
excellent
helps.
advice.
And also I think it would be really it wouldn't be complete if I don't ask you this question, because then our audience will probably be disappointed. If you're looking at an application, oftentimes students, an application, let's say, for a PhD, or let's say a master thesis student or something like that. I guess it's still at different levels, you look for different things. Let's look for PhD, which is, let's talk about PhD application, because that's one of the most, I think, if statistically the most applied applications from India outside. So if we talk about it, people oftentimes don't know whether they have to. So there are two aspects of it. They have a CV and they want to just sort of expand the CV. They have a checklist. So we add this. Versus if you can back it up, what you've written, by the skills. This we talk about an actual applicable skills, like skills in R, RNA-seq data, microscopy, whether that is important. So I know that has to be a balance, but I think oftentimes people get lost in just making a checklist on the CV. It includes publications as well, which it definitely helpful, but I don't think that should be the primary focus as to that. Having some substantial. What are your thoughts on?
As is a CV for a PhD is a very kind of skill piece of work, I think. It's and I think there are probably some, at least for me, so I'll talk from the point of view of what I'm looking for. A PhD student, a good PhD student, and often I'll talk to my team about, your CV needs to get on the yes pile. You don't want it on the maybe or the no pile, because the maybe no pile is probably not going to make it to interview. So your aim is to get on the yes pile, and to do that you need to kind of show actually an awful lot of different things. So you need to have good academics, you need to be bright enough to kind of fulfill the academic eligibility requirements for the country that you're applying to. But you also need to have some of those kind of upskilling things that we were talking about, some experience. And I completely agree with you, actually, some tangible explanation of how you utilize those skills practically can be really helpful, because sometimes what I see is that students will say, oh, I can do Western blotting, for example. And then you meet them and you ask them about their Western blotting expertise, and they say, oh, we did a lab practical. Like, okay, that's great. You've seen it through, but that's not quite the same as running an experiment for yourself all the way through, from like the hypothesis generation all the way through to the interpretation and the conclusion. And so that demonstration of those skills actually is really important. And so something that is really helpful, I think, to see on a prospective PhD student application is almost like a summary. It can be 3 or 4 sentences of what they've done, which shows what they wanted to find out, what they found out, and what they think it means. And that actually, those 4 sentences tell you a lot about the prospective applicant. They tell you that they can do the technique, whatever it is, that they can design an experiment, that they can execute an experiment, and that they can, really importantly, they can think about what it means, and they can articulate that to you in a scientific and concise manner. And those 4 sentences might be really difficult to write, to kind of get it down to, and that, 4 or 5 is not like a defined number, but, a short paragraph. But that can tell you a lot, not just about the practical skills, but also the intellectual thinking skills of the prospective PhD student. And actually, it's that potential, that latter potential around the thinking and the ability to critically appraise, to interpret data, which is the starting point of a PhD. You need to have the desire to develop that skill, because I think that is what a PhD is. A PhD is the training in becoming an independent scientist who can take the whole, the whole experimental design into the bigger picture. And we're not expecting you to be the fully formed entity at the application stage, but showing that you have the potential, I think, is really important. That was quite a long-winded answer, I think. A very nice, succinct question.
That's perfect.
I think there's quite a lot to think about in how you frame it.
I think it's a really critical advice, because oftentimes, and now that I've come to think of, this is beautiful advice, because I think this will really help concisely telling what you've done at a certain place in a project, or a course, or anything else. I think I think it is going to. I think it's better to do this rather than making longer CVs, which are. I mean, this is definitely going to be as long as it, but it's more substantial.
Right? It's a bit, and it's a bit more narrative as well.
Exactly,
and things end up being a bit more joined up, I think.
Exactly, it's beautiful. Great, thanks a lot. But the last question before we end.
Okay.
I think this is it. You don't have to stick to biology, but anything in this scientific world. If there was one discovery which gave you goosebumps, what that would be? Like, it could be anything which you saw recently. Most goosebumps or most, excitement. A discovery or such.
Oh, that's a really difficult one.
It could also be space. It could also be, I don't know, chemistry. It could be any subject. It doesn't have to be geography. Or in your field. Could also be in your field.
Yeah, I guess something. I mean, I think they're still working on it. But there was, and so I don't think there's a peer-reviewed article in it, but there was a marine vessel which goes around exploring the sea, trying to understand, 2/3 of our world that we know very little. We know less about the sea than we do about space, for example. And at the end of this voyage, one of the team who'd been on the ship notices kind of goo in the rudder of the ship. And so they collected the goo. Why not? Because they're scientists. And they took this kind of oozy black goo back to the lab, and they thought, why don't we just sequence it and see what's in the black goo? And because the rudder is a kind of, the area that they kind of got the goo from is not oxygenated. And they did this sequencing, and they found, unsurprisingly, a number of microorganisms that don't require oxygen to survive. But they found a whole load of brand new things that we never knew even existed from this goo in the rudder. And I just think that was, so they don't know what they do, they don't know what they are, they don't know what they mean. But I think that's just like a delightful example of science in action, where you see something and that curiosity we were speaking about earlier, where they're like, oh, wonder what this is. Let's go and find out. Why not? And that initial observation of the goo might well lead to, if we come back and chat in 5 or 10 years' time, might lead to some really fascinating discoveries about how organisms live without oxygen. So I guess, it might not be the most eloquent Nature paper, but I thought that was a lovely science in action story.
That's actually really cool and I'm really glad that you brought that up because it ties up to what we do in microbiome science. We exactly do that. Like
we've gone full circle.
Let's take this human sample and see what bacteria there are. So this is really cool. Thanks a lot for all your really great inputs, advice, and talking about your science. I think it's been an absolute pleasure.
Thank you very much for the invitation. It was lovely to meet you.
Lovely to meet you, and hope we keep in touch in the future.
Of course. Very happy to do so.
Thank you very much, and have a great day.
Take care. Nice to meet you both. I'm sorry I couldn't hear you very well, Manoj.
No problem.
Actually, my side is networking issue. That's why I hope so.
Okay, good.
Take care. Have a good day.
You too. Bye-bye.
Bye-bye. Bye.